Nucleoprotein. Shown above, nucleoprotein of a different virus. This is the Ebola virus, NP. Not the same fold at all. It doesn't look anything like our nucleoprotein, but there isn't a good three-dimensional high atomic resolution structure of the coronavirus nucleoprotein yet, despite all that has been done. So anyway, that's your mascot. All right, nucleoprotein probably looks like this. This is from a recent cryo-EM structure, and when there is a nucleoprotein in its natural form, it's going to be a dimer. So it has a C-terminal domain here, and that's also called the dimerization domain. That's the part that's going to link up. And then the N-terminal domains sort of fold around each other, kind of like that. That's what they're doing here. So the N-terminal domain just means it's the front domain that actually has a structure that has been solved. And so that's this blob up here. And here's the blob from the other monomer. And then in between the N-terminal domain and the C-terminal domain, you have the SR-rich region, which is this middle blob. And here's the middle blob here. And that is loaded with serine, amino acid code S, and arginine, amino acid code R. Yeah. That's why it's called that. At the end, there's a thing called the N3 domain, and N3 probably doesn't get to have a structure unless it's interacting with the M protein and or the genomic RNA, in which case it probably does have a structure. Nobody's been able to figure out what that is yet. I'll give you a little insight here in a second. So yeah, that's N. Two big domains that are always conserved, N-terminal and C-terminal, a sort of a blobby connector region that is super basic and polar in the middle, the SR-rich region, and a little tiny thing at this end that doesn't have a structure until it is doing something, and nobody's been able to catch it doing the thing that it does. There's also a little bit out at the front end that's not conserved, and don't worry about it. It doesn't even get a name. Yeah, gets no respect whatsoever. All right. So, a good question to ask now, and one that we forgot to ask on the M, is how well conserved is this N protein thing? And so you can see the N protein itself is present pretty much everywhere there's a virus. In a few cases where you don't get it, there's a little jagged line there, which means we're missing that part of the genome. We know it's not complete. Probably there is an N. So every coronavirus seems to have something like an N protein. Possible exceptions here, but they even have at least half an N protein, and maybe it's more than half an N protein. It's a real weird one. Don't worry about it. Yeah, red pandas and badgers are those weird guys. All right, so N protein. Let's say universally conserved. Another protein that's pretty much conserved everywhere is the M protein. That's this red one. And again, red panda and badger are being weird. Just ignore them for a minute. Yeah, don't give them your time. And you can see S is absolutely everywhere that is a coronavirus. E, which is our other structural protein, is the little orange box. When it's present, it's right in front of M. And so you can see it in this group and in this group. You can see it missing in most of this group, but here in about half of it. And you can see it missing in every single one in this group. So E is sometimes there, sometimes not. And so sometimes the virus can learn to live without it, or sometimes it gives it a little extra help. Yeah. But anyway, we're here to talk about N. N's doing fine. There's N. All right, so let's close that and that so we can see the multiple sequence alignment, because that's what you come here for. This is the good stuff. Let me move this over just a little. All right, let's go back to the start of N. So I tried to make an alignment of all the N proteins from all the coronaviruses here. Can't be done. Yeah, or at least I haven't figured out a way to do it yet that makes any kind of sense at all. N protein changes a lot. There is always an N protein, but it's not always the N protein that we're thinking of. So what I've got here are the alpha and beta coronaviruses. So this would be a group that includes SARS coronavirus 2. There, SARS-2, that line. So this is going to be the one that is most similar to the thing that we're going to become most familiar with. And so I wanted to look at all the ones that are closest to that. So like we said, at the beginning you got this little region where there's nothing conserved. Don't worry about it. And then as you go on, you're going to see a lot of conserved little red bars. Those are going to be K and R, the two positive charged amino acids. And you'll see some other stuff too. Yeah. So the first thing you'll come into is this region. It starts right here with these two red bars, and it goes all the way down to just about here, actually. This is the N-terminal domain, the amino terminal domain of the nucleoprotein. It's hard because it's the N protein and the N-terminus, but they mean different things. So I'll try to say amino terminal domain. How about that? And you can see this has a lot of things that are conserved. There's a big chunk in here with a whole bunch of, they're all aromatic amino acids, so they may be doing some kind of crazy pi stacking interaction. That's a thing that aromatics can do with each other, and nobody else really gets to participate. It's just their thing. It's their special little in-group thing. You see some nice conserved basic amino acids, and this domain will bind to RNA if you put it in the presence of RNA, but this doesn't appear to be the domain that gives you specificity for packaging the genome RNA. So this thing can't tell, messenger RNA from ribosomal RNA from whatever. Yeah, it just likes RNA in general. Great guy, the N protein, the N-terminal domain, amino terminal domain of the N protein. All right, and there's a little more. You see, you get that W running down there. That one also could be involved in pi bonding, pi stacking. Then we hit the Christmas lights, red and green, or if you're colorblind, gray and gray, the way Christmas is meant to be. You can see right here, we've got a lot of conserved arginine, a lot of conserved serine. This would be the arginine and serine rich region, or the SR rich region, serine arginine. If you look, I've pushed a bunch of these other things sort of together, sort of squeeze all the air out of the alignment, but most of what's there is either R or S or N, which is just polar. It's the thing that's not serine, but is kind of doing the same job. So yeah, this is a big old region that's going to add a lot of positive charge, give us a great generalized affinity for RNA, and is not going to be specific RNA binding at all. You're not going to get specific packaging of the genome from this section, but boy, is it a good clamp. And so this is going to be on the outside of the N dimer, and probably some of the RNA can wrap around that. All right, you have the mystery region. Don't worry about it. I don't know what that is. And then you have, this is called the C-terminal domain, or carboxyl terminal domain of the N protein. And here we have some things that are acidic and conserved, some things that are basic and conserved, and some things that are just sort of conserved. So this is a region that is going to do dimerization. Two of these are going to link together. And it's also going to use these conserved red columns, these lysines and arginines, seem to be the ones that give us a specific genome packaging. They're going to be arranged in kind of a ring going around the outside of the dimer. And yeah, the data looks pretty good that these are the thing. Yeah, so there is the C-terminal domain. You can see it's well conserved. Then there's the region of chaos. And then this little thing, these little purples and blues, those are, as best I can tell, the N3 domain. So these things are acidic, so they can interact with basic things. And so there are some conserved basic residues that you would find in the tail of the M protein of some of these viruses, and it may be that they're interacting there, or they're interacting with, I don't know, it doesn't seem like something that would interact well with RNA, but that's your N3. So it's a really dinky little thing, and yeah, no wonder there's no structure for this. What you've got, though, is polar, hydrophobic, polar, actually, acidic, hydrophobic, and acidic again. So you've got a 1, 2, 3, 4, 5, 6, 7, 1 arrangement, which to me looks like an amphipathic alpha helix, with your hydrophobic side being the thick side and your acidic side being the thin side. So potentially that's the shape that it's going to make someday when somebody solves this structure. And you heard it here first. So that is the nucleoprotein. Now if you look at other nucleoproteins of other coronaviruses, other nidoviruses, Sometimes they are missing the amino terminal domain. That sometimes just goes away with all those exotic interactions in there. There's usually something like the arginine serine rich region, although sometimes it's an arginine glycine rich region, which no big deal, no big difference. The thing that tends to be conserved is the C terminal domain, this part. But even that is not well enough conserved that you can be absolutely certain that you are looking at the same thing. It's one of these things that just on the fringes of what we can detect with the most powerful homology methods. So it would appear, once again, there are a lot of possible solutions to this puzzle. Nidoviruses and coronaviruses are exploring all of them, and there are a number of them that work, and you can only really recognize it as the same solution when you're talking about viruses that are pretty closely related, like alpha and beta coronaviruses. And as for how they interact with M, yeah, alphas and betas probably do it in the same way. Does every other coronavirus or nidovirus do it in anything like the same way? Who knows? Yeah. We'll be finding out together by reading the literature or doing the work in labs. So there you go. That's an overview of the nucleoprotein and its important domains and what they're there for. Thanks.